Спасибо большое.
Все готово.
Good afternoon, guys. I see that you start coming to listen, and good morning, everyone. Today we'll discuss bacteria that's supposed to be potential agents as biological weapon. So, why it is so? Because they are causative agents of dangerous infections. So, and we have many lists. If you take a look at the internet, we will have many lists of those agents. There's potential warfare agents. But in a short list out of those agents, we 100% will see the names of Yersinia pestis and the disease plague it causes, Rickettsia provazekii that may cause epidemic typhus, Borrelia reccurentis that causes louse-borne disease that is called epidemic relapsing fever, Vibrio cholera that may cause cholera, and of course, the last but not the least, Bacillus anthracis that causes plague. Of course, you have to understand that it's not possible today to discuss them all in details like we discuss it in a class. I just give you the general overview of the pathogen and the disease it causes. And from the very beginning, I would like to tell a few words about the agents that can be considered as biological hazard or biohazard. And you can see the sign. So on the yellow field that supposed to be a sign of biohazard. And what are biohazard, or what is biohazard? This is a biological substance that poses a threat to the health of living organisms, primarily humans. This could include mainly microorganisms, bacteria, viruses, or toxins produced by bacteria or fungi that can adversely affect human health, and biohazard also could affect animals and can be also biohazardous to animals. So traditionally, we divide all microbes according to the severity of the disease, the properties of those microbes, into several biohazard levels, more commonly referred to as biological safety levels or biosafety levels. This classification is based on the safety precautions that should be undertaken to in clinical or research laboratories, depending on special pathogen handles when performing laboratory procedure, and regarding the contagiousness or infectivity of the microbe, the severity of the disease it causes, whether it is easily transmitted from the source to the susceptible individual or not, whether it is easily treatable or not treatable, and whether the specific prevention is arranged or not. So traditionally, in the American classification, the fourth level of biosafety is the highest needs highest isolation of the laboratory from the environment, because the most deadly organisms usually belong to level 4. So, and in that level, out of the organisms, we have, for example, Ebola and Marburg infections. So the rest that we mentioned today that are Yersinia pestis, Bacillus anthracis. By the way, also the one that we don't mention here, but this is the important microbe, HIV and some other viruses, they all belong to the level 3. But generally saying, the very high level of isolation is needed for the laboratory, clinical or research laboratory, to handle this microbe. And by the way, coronavirus also belong to level 3. So now we take a look at the particular microorganisms. And of course, we start with Yersinia pestis, the causative agent of plague. Because the plague is really very serious bacterial infection. And unless treated, the lethality may make 50% in one of the most common clinical forms, that is bubonic plague. So the mortality rate will be around 50%. And if we have pneumonic plague in population, the mortality usually makes 100% if non-treated. But by the way, even if treated, bubonic plague may give lethality up to 10 to 5%, and pneumonic plague, even if we have some delay in treatment, may reach the lethality rate 50 to 30%. So pneumonic plague is a very severe state of the body. You can see here that natural source of infection is the wild or domestic rodents. They are most susceptible for the disease, but carnivores also can serve as a source of infection. From animal to animal, the disease is transmitted by a flea, so blood-sucking insect. And when the insect is infected, it start biting not only animals, but if humans occasionally enter the environment where these fleas occur, they may bite humans also. So we will see the formation of the most well-known and famous symptom of the plague, that is bubo development, inflammation of the inguinal lymph node. So we'll mention it later on. So, few words about the microbe itself. Take a look at the microbe. That is the scanning electron microphotograph of the microbe. And here the staining to see the very typical so-called bipolar staining. You can see here that this organism is the small, sometimes pleomorphic rod. The Gram stain reaction is Gram negative. And you can see here several names. So initially they were classified together in the same family, Yersinia, Francisella, Pastorella. Now Yersinia is classified into Enterobacteriaceae family. And the common features of this were they have animals as a natural host, so they infect them naturally. But then all three could cause diseases in humans. All three look similar, than the gram-negative pleomorphic rods. So the basic enzyme that help to differentiate their genera, that they are oxidase negative and catalase positive. And due to oxygen requirements, they are facultative anaerobic bacteria or microaerophilic. But then let's take a look at the history of the disease they cause. So within the history of the mankind, we could see at least 3 very important pandemics. I would like to draw your attention and to remind you that what is the difference between epidemic and pandemic? Epidemic usually occurs within one country, so pandemic usually occur within continent, spread within continents. And if you take a look at the history of the plague, the first very well described pandemic occurred in the 6th century AD. And within this, around 100 million people had been killed within the 50 years within the epidemic. The second major pandemic that got this name, Black Death, originated in Asia in 14th century, and then from there it had been spread to the Near East and Europe. And within 15th 16th century, in Europe alone, 1/4 of population of Europe died of this disease. So it was really devastating pandemic. And the third pandemic originated in Burma in the late 80s, and it spread to Chinese, and from there it was spread almost to all continents, and spreading occurs via rat-infested ships. So, and it led to the establishment of natural foci or focuses of the disease and wild nature in almost all continents. Now we have the natural foci in Africa, North America, South America, Russia, and a lot of other countries. The most devastating pandemic occurred in the 16th century. And you can see here the picture of the great artist, Peter Bruegel, who showed the on his picture that is called the triumph of death. So he demonstrated the fear that occurred and the feeling of unescapability from the plague. Because you can see here the king at the very corner here, dying on the street. And you can see the monks in a carriage, those that take care of the corpses, burn them. And you can see dogs eating corpses on the streets. And you really can see the terror and fear in the whole picture. That's how people thought of the plague in the Middle Ages. So, how about the plague today? And it's not just historical disease. So the thing is that every year we have some sporadic cases in some of the countries. And here you can see the result of the WHO from 2010 and 2015. And altogether within these 5 years, there were 3,200 cases reported worldwide, and out of them, more than 500 people died. And you can see here that the red one is the cases, and the pink one is the cases, and the brown one, the proportion of mortality of death cases within it. So, and with red color, here it is indicated the potential natural focuses are indicated that occur everywhere within the world, but then you will pay attention that mostly we have them in Madagascar and in Congo. So every year, as I said, we have some sporadic cases. For example, not very far ago, around a month ago, the new cases emerged in Mongolia and China, on the border between Mongolia in the province Inner Mongolia and China, and on the border between it and Russia. So, and it happens in July 2nd, where death occurs of the people who are hunting and eating the most common natural hosts of the plague. They are marmots. And since that time, and you can see a picture of the boy who died out of Yersinia pestis infection after hunting these marmots. So, and since that, so this meat of this very nice animal, pretty and cute animal, considered to be delicious in Mongolia, and usually they are hunting them. So then it is prohibited since the appearance or outbreak of the disease there. So it means that theoretically, it is not possible to eliminate the natural foci of the plague where it occurs naturally in wild animals. So that's why we have to supervise and monitor and the ability of the disease to spread out of the natural forces, and mainly from animals to humans, to be transmitted from animals to humans. Few words about the morphological and cultural features of Yersinia pestis itself, not the whole family or group of bacteria, but Yersinia pestis itself. They are quite small rods, non-motile, non-sporin, and they are rods and they are gram-negative, but usually when we take a specimen from the patient, and you can see here there's an image smear of the lymph node. What does it mean, image smear? If we take the inflamed lymph node and touch the slide and then fix it and stain it, so but the staining here is with methylene blue staining, so we could see the organism or shadows of the organism. So you can see the so-called bipolar staining. The poles are dark and the middle is light. This is a scheme, simplified scheme of the bipolar staining, and that is true picture from under the microscope. So Yersinia pestis have particular features, cultural features or growth features. They are really very sensitive to heat, sunlight, drying, chemical disinfectants, but then they are resistant to cold in moist environments. So they may survive in that environment for a very long while. And also they stand freezing, and they may resist freezing in the infected fleas, for example, and they also remain viable for several months in the soil of rodents' burrow, and for half a year they remains viable in the organisms of not frozen but alive fleas. So what are the growth characteristics and how are we going to culture them? So it is not fastidious organism. It may grow on almost any media. Could you imagine if it survives in the rodents' barrows, right? So then, of course, they may grow at very simple conditions on very simple universal medium as NPA. And Yersinia pestis is really famous for its wide or broad range over the temperature it may live. So it stands the temperature from 2 to 45 degrees Celsius, and it is known over a wide range of pH. It may grow from 5 to almost up to 10 alkaline medium. So they grow faster in 30 degrees Celsius, and for as a selective medium, McConkey's medium will be used, and it forms colorless colonies of McConkey's medium that differentiates it as lactose negative, not able to ferment lactose. So when we culture it on blood agar, the colonies start to be dark brown because they absorb hemine pigment, and that's important because they may get it in our own expense from the body. So, and if the colonies of Yersinia pestis are viscous, so it means they are highly pathogenic because the only virulent strains produce this type of colonies. Yersinia pestis may be an example of the highly pathogenic organism with many virulence factors. They really demonstrate the very long list of virulence factors. Of course, as an gram-negative organism, they have lipopolysaccharide, and its lipopolysaccharide is really very potent. So then it is covered with the envelope protein that is called fraction 1, and it makes the antiphagocytic heat labile envelope of the protein. So, and also it is called fraction 1 antigen. So it is interesting to mention that almost all virulence factors of Yersinia pestis are encoded by the plasmids. So this one is encoded by P stands for plasmid, fra plasmid. This is a fraction 1 plasmid. So then, next, V and W antigens, they are also encoded by a plasmid, BCAD plasmid. And the plasmid is the plasminogen activator gene. And V and W antigens, they are important factors that may inhibit the activity of the complement system, inhibit phagocytosis. And these are very important virulence factors that help Yersinia to survive in the body and not to be eliminated by lytic or protective mechanisms of the body. Then they also may produce one very interesting factor that is called coagulase. The thing is that plasma coagulase is produced in the flea body but not in the human body, because the temperature suitable for when this property will be expressed and coagulase will be produced is not more than 28 degrees Celsius. So it means the synthesis of plasma coagulase at a temperature higher than 28 degrees Celsius will be blocked, so it will not be produced in the human body. And this property make possible transmission of the disease by the flea bite from animal to animal and from animal to human. And this transmission will be lower in hot weather because temperature of the flea will be too high to produce coagulase. So, and again, the coagulase enzyme is encoded by pestis plasmid. Then they also may produce fibrinolysin that is encoded by the same plasmid. It degrades fibrin clot and permits Yersinia to spread rapidly in the body. So if it is trapped in a blood clot naturally. Then apart from this, they may have a lot of so-called YAD proteins and YOP proteins. I cannot see yard proteins. So yard proteins, YAD proteins, these are the factors of adhesion. So yard proteins help bacteria to bind with the membrane. But then they also may produce a lot of so-called YOP proteins, Yersinia outer membrane proteins. And they are usually components of the type 3 secretory system. Do you remember I said about this system, I think, in the general microbiology classes? So then, this system, this is the type of the pili or secretory pili that make a special syringe. You can see here this is a bacterial membrane and this is human membrane, and this is the potential scheme of it, and this is microphotograph of this type 3 secretion needle or type 3 secretion pillars. So some product, toxic product produced by bacteria will be directly injected through two membranes, bacterial membrane and human membrane, directly in the human cytoplasm. That's how outer membrane protein that are cytotoxic can be delivered directly from bacterial cytosol to the human cytosol, and also some toxins can be transmitted like this. So we think that this family of the YOP proteins are very important because they code for type 3 secretory system, cytotoxic proteins that also inhibit the activity of macrophages and platelet segregation that may prevent the spreading of the microbe. So some more factors can be mentioned. For example, pestis, that is a bacteriocin produced by Yersinia pestis. And we have discussed already that bacteriocins are the substances not dangerous for humans, but dangerous for some other bacteria. So pesticins may inhibit the activity of E. coli, may inhibit the growth of other Yersinia, so they may give Yersinia pestis selective advantage to colonize the particular surfaces and to survive in particular ecological niche. Also, we may mention the surface component that can help Yersinia pestis to absorb organic iron or hemine from the red blood cells. They also may produce typical exotoxin. It is called murine toxin because it may kill small rodents very quickly. Not all rodents are equally susceptible for it, mainly mice and rats. So, for example, guinea pigs and primates are not susceptible to this toxin. So, it is called fraction 2 antigen. Do you remember fraction 1 is the envelope protein and fraction 2 is the internal murine toxin that is thermolabile protein that can be toxoided. So, and you can see here some extra virulence factors, right? So, these are the, okay, so we discussed that is the type 3 injected system. So, where Yersinia may inject directly the products, your protein that are cytotoxic, into the human cell. This is a human cell. And these outer membrane proteins may activate different signaling mechanisms that will send the signal from the outside the macrophage into the nucleus, and then they start cytokine production. This pro-inflammatory response will be produced, and this signal may inhibit the apoptotic signal of the macrophage, so macrophage survive for a long while and may shed Yersinia pestis and so for Yersinia pestis multiplication. Few words about clinic and pathogenesis of the disease. Clinically, based on the symptoms of the disease, we may categorize them into three forms: bubonic, pneumonic, and septicemic. So you see that bubonic plague comes from the flea bite. But then as soon as we have bubonic plague and bacteria enter the bloodstream, may spread through the bloodstream, and they may cause different diseases, including the disease when they may enter the lungs. So that will be pneumonia, and that will be pneumonic plague. But because the initial site of entry of the microbe was different, so this pneumonic plague is called secondary pneumonic plague. And during this, the host organism, macroorganism, the human, start releasing Yersinia pestis from the nasal and secretion with sputum coughed out of the lungs. So, and then it means that this person start to be highly infectious and may spread Yersinia pestis through sputum. Then it means it may directly transmit it to the susceptible individual without biting of the flea. Then the one that will be infected will got primary pneumonic plague because the one will get it through the inhalation of the infected sputum. Septicemic plague is when microbes spread through the body and infect almost any organ, so they may cause pneumonia, hemorrhage of internal organ, so it may spread through lymphatic system, so and it may even cause the gangrene of the distant regions of the body due to disseminated intravascular coagulation in the blood. So, pathogenesis of bubonic plague is the most common disease caused by Yersinia pestis. Usually, flea feeds on the rodents, and if they're infected, with the blood they suck in the bacteria. So bacteria multiply in the blood in the organism of the flea, but then bacteria produce coagulase, and then it blocks the preventriculus of the flea. Then food, blood cannot enter the enteric system of the flea, so flea feels hungry. This flea is called blocked flea because the preventriculus is blocked and it feels hungry. As far as it feels hungry, it starts biting without any choice. It starts biting animals or humans if they enter the area. So that's how disease can be transmitted from animal to human. So when it bites a human being, the aspirated contaminated blood can be regurgitated into the biting site, and the one will infected through the wound, and the ulceration will occur in the biting site. So, and the inoculated bacteria cause inflammatory response. Their monocytes and macrophages come up to the area and engulf it. Since they engulf it, macrophages start moving them into the lymph nodes. And you can see here a very old picture. So when from the middle ages, when the doctors collect together by the bedside of the patient with the bubonic plague, and you can see the inflamed lymph nodes in the body, right? So usually the most commonly inguinal lymph nodes start to be inflamed because macrophages bring Yersinia pestis there, and they are named bubo. So the disease had been named bubonic due to the bubo, or meaning growing. And hemorrhagic inflammation of these bulbs occur, then necrosis of the lymph nodes. And the contact with these necrotizing lymph nodes will be highly infectious because Yersinia pestis will be released from these bulbs. And also this shows that dissemination through the bloodstream occurs, and usually it leads to sepsis and gangrene of the skin, fingers, penis, meningitis, pneumonia, and death. And a few words about the epidemiology of the plague. Usually, we may distinguish two cycles: sylvatic cycle and urban cycle, where microorganisms may occur independently. So usually they occur in wild rodents, and the wild rodents are infected through the flea, and one flee infect the other one. But also not also the rodents may participate in this. For example, carnivores are also susceptible to disease. So due to the flea bite, the carnivores can be infected, so they maintain this wild or sylvatic cycle also. So it's the same we have in the urban cycle. So because there are a lot of domestic rodents, or rodents that live nearby the villages in urban area. So then also the rodents and domestic carnivores may maintain this cycle. But from either of those cycles, if a human being enter this area, so they can be infected by the flea from one or another cycle, and since they are infected and the pneumonic plague occur, the cycle will occur in humans only, and can be transmitted without fleas, because pneumonic plague can be transmitted directly from one person to another one. So this is like a third cycle of Yersinia pestis. So how we usually diagnose the disease? So microscopic examination is of high diagnostic value if you demonstrate bipolar staining of the organism, but this is not enough. So usually we also perform direct immunofluorescence strain, and to confirm the presence of Yersinia pestis surface antigens. Usually fraction 1 antigen is demonstrated. But then, according to the regulation of WHO, the culture should be performed. But of course, the culture of this microbe can be performed in any laboratory. Of course, it can be performed in a laboratory with safety level 3, with very high degree of the laboratory isolation from the environment, not to proliferate bacteria and to contaminate the environment. So, and all cultures must be handled with extreme caution due to the very high infectivity of this organism. And usually blood agar, McConkey's agar is used, or blood agar, and they don't produce they cannot ferment lactose on McConkey's agar, the colonies will be colorless. So sometimes we may confirm the diagnosis by serological tests, and for this we may use passive hemagglutination test or complement fixation test. And usually we have to confirm that this disease is zoonotic. So that's why usually the animal inoculation is performed. How about treatment? Of course, Yersinia pestis is susceptible to many of very good, old good antibiotics, streptomycin, tetracycline and others. So, and you can see the drug resistance in Yersinia pestis is rare. But by the way, my dears, we now understand that Yersinia pestis organisms, if needed, may be easily genetically modified to be more resistant to the drugs. And that can be performed as a result of genetic engineering, and that make the Yersinia pestis a very possible potential threat when using as biological weapon. Plague is generally preventable disease because first, for prevention we may need control of fleas and rodents, and this is general prophylaxis, general measures. But for specific prevention of the disease, live plague vaccine, you can see here, is developed from the particular strain for single subcutaneous administration. So it means from one single shot the immunity will be developed, but unfortunately, so the vaccine cause not long-lasting immunity. Why? The level of antibodies will be seen, but the disease is so severe that very high level of antibodies is needed to prevent the disease. So, and this very high level of antibodies will be seen just within a year or 2 years. So it means that this vaccine can prevent the bubonic plague development, but it unfortunately cannot prevent pneumonic plague development. So, and usually the vaccination occurs in those who are at risk of contacting with the disease. What are they? Who are at risk? Of course, forester rangers that may contact wild infected animals, or people who are working in the laboratory workers, or maybe some rangers that are sent for some special operations during the war to other areas, something like this. But of course, this vaccine is not for everyone, but theoretically the stock of vaccine can be done in case a large scale use of the vaccine to prevent population from being infected with Yersinia pestis. So this is enough for Yersinia pestis, and then we switch to another organism that is from absolutely different group, and this group of organism is called Rickettsia, and it is called by the name of the doctor Ricketts, who discovered this bacteria, studied this bacteria, and died of this bacteria. So, what are the general properties of rickettsia? And I would like to remind you that this is not normal, traditional, or regular bacteria. They are different from regular bacteria because they occur somewhere in between bacteria and viruses. It means they have some features of the viruses. So names of these bacteria, or at least genus, of these bacteria: Rickettsia, Orientia, Ehrlichia, and Coxiella. These bacteria initially had been placed in one single family, Rickettsia, but then since that time the taxonomy of these organisms had been rearranged and fixed many times, and still the taxonomy is not settled yet. So we will consider them all together as the related genera. So, but then we'll talk more about Rickettsia themselves. So, all those four groups of organisms are aerobic, gram-negative bacteria, very small, and according to the shape, they are cocobacilli. What feature of the viruses do they demonstrate? They are obligate intracellular parasites. What does it mean? It means that to develop themselves, to multiply, they, they need to enter the cell and develop themselves inside the cell like viruses. They cannot grow outside the cell. They need a living cell to grow. Apart from this, that can be transmitted through arthropod vector. So that's why they may occur as a part of a natural cycle. So not all of them may need arthropod vector. For example, Coxiella may be transmitted through the milk of the livestock or goats, if it is not boiled or pasteurized. Now, let's take a look at the structure and physiology of this rickettsia. So, all those bacteria that we mentioned, all for genera, they have cell wall. And if you compare it with the cell wall of other bacteria, it resembles the cell wall of gram-negative bacteria. So we have peptidoglycan layer, but this layer is very thin quite often, so not fully developed. And then, as in gram-negative bacteria, there is an LPS there, but endotoxic properties of LPS is very weak. So, because this is a very thin and not very well developed layer. So the polysaccharide layer is slim and small and thin. Then they also may have fimbria or pili that may help in conjugation in between F positive and negative bacteria that serves for exchange of genetic information within different organisms. So these are non-motile bacteria. So, and I would like to underline they are obligate intracellular parasites. They grow in eukaryotic cells also. These bacteria are quite small. It makes from 0.3 to 2 micrometers in length, and they can be hardly stained by gram staining, very poorly stained by gram staining. So, culture method is not really used in diagnosis of this disease. You should understand those reasons. First, they are very fastidious and they can be cultured like viruses in guinea pigs or in animals. So, this is animal inoculation. If you take blood sample from the patient, clot it, make a clot, and then inoculate guinea pigs, tissues, or embryonated hen's eggs. And do you remember we mentioned that yolk sac can be used for Rickettsia isolation. But because these bacteria are very dangerous and hazardous, very few laboratories may perform isolation of these bacteria for diagnostic procedure. And to work with these bacteria, safety level 3 is needed, so almost the highest level of isolation of the laboratory from the environment. And uh, quite a lot of cell cultures can be susceptible to rickettsia, so it is cultured in a way as viruses can be cultured. So what diseases are caused by common rickettsia? So according to the diseases, those rickettsia may be classified into several groups. We mention at least two groups. These are typhus group and spotted fever. Due to the most important symptoms. Typhus group, the most important symptom is a very high fever. Spotted group, the very important symptom is the hemorrhagic rash along the body. So, and in typhus group, Rickettsia provazekii, that causes epidemic typhus, of our today's interest, is a louse-borne disease and is transmitted with feces, louse feces, and it is only if it epidemic, so there's an anthropootic infection that spread through humans only. While the endemic typhus that similar with the first one by clinical symptoms, it is transmitted by flea feces and the natural hosts are rodents and the humans are the occasional hosts of this bacteria. Then the second group is spotted fever group. By the way, Rocky Mountain spotted fever is the first rickettsial disease identified by researchers. And in this group we have Rocky Mountain spotted fever, and this is discovered in the USA first, and then some other diseases. So they are transmitted all by biting of ticks or soft ticks. So, and for spotted fever, the natural hosts are rodents, or for others, rodents or dogs may be the hosts. So, what is the pathogenesis of rickettsia infection? The question is how rickettsia may cause harm to a human body. So when rickettsia enter the body through usually biting site over the louse or tick, they enter the body through the biting site and target cells of monocytes and phagocytes for them, and they engulf these bacteria or internalize them. So, when these bacteria internalized into the vacuoles, they easily escape from vacuoles into the cytosol. So that's how they escape digestion by phagocytes. So this escape is mediated by the enzyme that you also may find. Similar enzyme may find in many bacteria, including Staphylococci, for example. That enzyme is called phospholipase, and this is a pronounced virulence factor of rickettsia. So, rickettsia may multiply in the cytosol by binary fission, and of course, they cause damage to the cell. They occur where they occur, and usually they cause the damage or harm to the cells directly, and then they can be spread from one cell to another one by actin mobilization. And you can see here that we can on this photograph different stages of the pathogenesis: attachment, phagocyte or internalization of the rickettsia, and rickettsia escape into the cytoplasm, so where they are not in vacuoles. And you can see that binary fission, so how they multiply in the cytosol of the macrophage. So in rickettsia, the degree of the damage that they cause to the cells is different in different types of rickettsia. So this is rickettsia in the multiply in the endothelial cell, right? So some rickettsia, like Rickettsia provazekii, are very hard. Act very hard on the cell. They rupture the cell. So the release of Rickettsia provazekii causes the rupture, the lysis of the human cell. But then we may have rickettsia that bond of the cell, so the cell may survive for a long while, and the severity of the disease caused by this rickettsia will be lower than that caused by Rickettsia provazekii. And also we may have rickettsia from the typhus group Rickettsia that may cause philopodia formation and focal lysis of the cell. By the way, the cell gonna die anyway sooner or later. So let's take a look at the disease, epidemic typhus, and few words about the history of the epidemic typhus. The first description comes from the 1083 from a convent near Salerno, Italy. But then now we know that most severe epidemics of epidemic typhus usually occurred during the wars and famines. So I would like to give words about the Napoleon. One could say that lice have defeated Napoleon's armies. Moreover, their retreat has spread the infected lice and typhus throughout the entire continent over the Europe. So, and that was about the war, when biggest epidemic of epidemic typhus occurred during Napoleon's retreat from Moscow in 1812, when more French soldiers died of typhus than from Russian bullets. And of course, during and immediately after World War I, that occurred in 1918-1920, 30 million cases occurred within the troops, and around 3 million deaths occur. Here you can see, epidemic typhus is caused by Rickettsia provazekii, and usually all typhus diseases caused not by rickettsia. We have some more, then we will know more about them in the classes. Usually it occurs, the onset is very abrupt, and the incubation period of epidemic typhus is about 14 days. So, and usually in typhus, the most prominent clinical manifestation is abrupt fever, very severe headache, abdominal pain, and some other effects, including effect to the nerve system, arthralgia, delirium, coma, and seizure. The disease is fatal in 10 to 30% of cases. And that's how the primary acute disease caused by Rickettsia provazekii appear. But appears that Rickettsia provazekii, after the initial disease, may cause persistency in the body. So it may not show itself for decades. But then when immunity is lowered, Rickettsia may start multiplication again to cause so-called Brill-Zinsser disease. This is recrudescence typhus. and that is the reactivation of previously occurred in the body Rickettsia provazekii. So the clinical symptoms of this disease are much milder than the symptoms of the primary epidemic typhus disease. So how do we diagnose epidemic typhus? Usually we perform microscopy, and because they are not really stained very well by gram staining, we perform Giemsa staining or immunofluorescence staining. Then we also identify antibodies, usually immunoglobulins M, because these are the first antibodies produced during the infection. Then culture can be performed in the reference labs only and is not used in routine diagnosis. Treatment and prevention. So this disease can be treated with all good antibiotics. They are very effective because these bacteria don't develop really the resistance against the drugs. So, but delousing is very important and usually essential in the management of epidemic typhus outbreaks. So whole phenol-killed vaccine is available, and attenuated live Rickettsia provazekii vaccine is also available, and of course they are not frequently used nowadays. So that's what I wanted to tell you about the epidemic typhus and Brill-Zinsser disease. So we shift to another disease. My dears, do we still have some time? Okay. So we have 40 minutes more, and 2 more bacteria. I understand that you may get tired because of the many bacteria discussed today, but just we take a look at the review of this bacteria. So we go for one more group of bacteria that we will study within our classes. These are spirochetes. But today we won't discuss all spirochetes. We'll just touch the one that is circled with the red circle. So this is the microbe from spirochete order, and it is Borrelia reccurentis. This is a disease that causes epidemic louse-borne relapsing fever. The others, they don't cause epidemics, and today we are talking about those that can spread through humans very fast, for example with the help of fleas or with the help of lice, and may affect many humans, and the diseases are very severe. So only those are of interest today. So Borrelia reccurentis may cause disease that is called relapsing fever, epidemic relapsing fever. The only humans are hosts for this Borrelia, because they are transmitted, the Borrelia are transmitted by body louse pediculus humanis. What do we know about these Borrelia? They are gram-negative bacteria according to the structure of their cell wall. According to the oxygen requirement, they are microaerophilic, and they are very fastidious in their nutritional requirements, so they can be hardly cultured on bacteriological media. So the morphology is typical for the spirochetes. They are helical rods. They are very thin, 0.2, 0.5 micrometers in diameter, but they are very long, up to 30 micrometers in length. So, and they are made up of 3 to 10 loose spirals. So, and there how you can see, this is electron microphotograph, and that's how you see Borrelia reccurentis in the blood of patient with relapsing fever, and the staining is Giemsa staining. We don't use gram staining for the visualization. So what is the structure of this bacteria? And please take a look at this slide, because this is explanation over the structure of all other spirochetes. They have similar structure of the body. So they are of helical shape, and it is imparted by the periplasmic flagella. So if we perform cross-section, that is cross-section view, you can see that this is periplasmic cylinder and this is a periplasmic flagella. So flagella insertion point, usually these we see bacteria laterally, is or are located near the termini of the spirochetes, on that side and on that side. And bundles of flagella fixed here wind around the flexible rod-shaped protoplasmic cylinder, and from outside they are overlapped, and they are overlapped in the middle, and from outside the outer membrane covers them, and as you see here, the flagella don't protrude to outside the bacteria. They are endoflagella. So, and they are covered from outside with a part of the cell wall, outer membrane. Do you understand? I hope you understand. And this is outer membrane. There is a flagellum that is arranged around the periplasmic space, and here you can see peptidoglycan in the cell, and that is the cell membrane. So how about the disease caused by this Borrelia? And I would like to repeat, that is called epidemic relapsing fever, louse-borne. After the person is exposed to infected arthropod, Borrelia enters through the biting site, and it doesn't usually multiply in the biting site. It goes directed to the bloodstream and spread to the multiple organs. And it multiplies there in the organs, and it is followed by a massive spirochetemia. What is it? Spirochetemia. When Borrelia multiply, they enter the bloodstream. So the presence of Borrelia in the bloodstream is called spirochetemia. So, and then spirochetes invade the endothelial cells or blood vessels, also multiply there. Once they are released into the bloodstream, in the course of the disease, we can see the first septic febrile episode. So what does it mean? Temperature comes up. So that is the picture of the first episode. See, that's what happened. We have primary fever, and it is very high. And this primary episode may kill the patient, may be fatal, because it is very severe. And it lasts for several days, from 3 to 6 days. And then within this period, the endotoxin is released. So that's why at the end it may cause hypotension and shock. So if the patient survives this episode, then it will end with the development of the antibodies. But the problem is that these Borrelia are able to change their surface protein's antigenic structure. So it means Some bacteria are killed already with the antibodies, but some survive, shift their antigens, and start multiplying again. So once they multiply and released into the bloodstream, so endotoxin will be released due to the lysis of bacteria. So we have second febrile episode. So from those bacteria that change their antigenic structure and survive the first attack of the antibodies. So there is a second relapse. So then, okay, it ends with several days, and we have second febrile episode. And because of the many relapses may occur, so this disease is called relapsing fever. But then the question is, when it's over? So Borrelia cannot keep changing the antigenic variants forever. They have several possibilities to rearrange the gene that code for outer membrane proteins. So then, when after several antigenic changes, Borrelia structure will revert to the original antigenic structure. So what will it mean? It will mean that in the body, the whole repertoire of antibodies will be produced against all possible antigenic variants of Borrelia reccurentis. Then it will be over because the antibodies are created against all possible Borrelia there. Then if the patient survives usually the first episode, then the patient may recover. So, and you can see here the very severe symptoms of the disease. So, and that's why that can be highly lethal. And you can see here that with colorful antibodies that first the antibodies are produced against the first variant, then against the second, then against the third, and so on. So the disease can be severe and death may occur in 10 to 40% of symptomatic cases in the absence of the treatment, but still if the patients are treated. So the disease may reach 5% in patients. And you can see here the historical photograph, how the epidemic occurs, and during the First World War the soldiers look for the lice to remove them and to prevent the spreading of the epidemic typhus. It was really very typical for the First World War, 1918. So then this disease tends to occur in epidemics. And usually it may occur not only during the wars, but for example, you know that some refugee camps had been formed in Europe not so far ago, as far as refugees start moving from other countries to Europe. So the outbreaks of the epidemic typhus had been found there because of the very crowding, high level of hygiene and sanitary, and usually they may also occur due to the natural disasters like tsunami, floods, when a lot of people are collected together in very poor sanitary conditions. Okay, I think it is enough. So the diagnosis of this disease occurs due to visualization of spirochetes in the blood during the febrile episode. Do you remember febrile episodes? Because within this febrile episode, we have the highest concentration of bacteria antigens in the human blood. So if blood is taken here, if blood is taken during the febrile episode, you will not see what we need. But if during the febrile episode, We may perform Giemsa staining of the thick or thin smear of the blood, and we can see tiny spiral-shaped bacteria in between the blood cells, red blood cells and white blood cells. So acridine orange can be also used for examining the bloods and tissues, but also Giemsa staining is performed. Because of the antigenic changing of the antigenic structure, serological tests are of very low diagnostic value and unreliable, so we don't use them for diagnosis. So right now the test kits for PCR are developed for these bacteria, so it is possible to diagnose them by PCR. The treatment is performed with tetracycline, penicillin G, erythromycin, and some others. Vaccine is not available for this disease. I hope you understand why. Again, we blame the antigenic variability of the surface structures. So they may change it, so the antibodies produced as a result of vaccination may not match with or their antigenic variants of Borrelia reccurentis. Now the next one is Vibrio cholera. Just briefly, so take a look at the electron microscope photograph of these bacteria, and you can see that they are curved and monotricate with one single flagella, and this is how we see them under the conventional microscope due to the gram staining, and they are a bit curved gram-negative bacteria. Very small, but they cause the potentially epidemic and life-threatening disease, and the major symptom of this disease is secretory diarrhea characterized by numerous voluminous watery stools often accompanied by vomiting, and this result in the hypovolemic shock, acidosis, and death of the patient if we don't help the patient with because very quickly the patient start to be very weak, unable to even drink him or herself. So that's why it is very important to take care of those people who are infected. So you can see the description of the organism, the polar flagellum, and that's why it is highly motile. So the oxygen requirements is what is true about Vibrio cholera itself, but not other Vibrio. It is strictly aerobic bacteria with very pronounced and developed fermentative properties. It grows within the broad range of temperature, not that broad like Yersinia pestis, that is broader, but from 18 to 37 it may grow. And it may stand the very high pH up to 9.5, and this ability to grow at very alkaline medium is used to develop selective medium for primary isolation Vibrio cholera from environmental samples from water, for example, or from the cases. Vibrio cholera is very susceptible to environmental conditions like heat, drying, acidity. So then they are very sensitive to stomach acidity on gastric juice. So it may not cause disease in very healthy individuals with a normally lower pH over the stomach. Normally, naturally, it resides or in fresh waters or brackish waters, and because of this, it can survive for months in sterile sea waters and in tap water. So that's why the water somewhere can naturally harbor Vibrio cholera. So we have many Vibrio cholera organisms, so that's why how to classify them. The classification is performed first based on antigenic structure. All Vibrio cholera organisms can be generally classified according to the structure of all somatic antigen into 139 serogroups. So, and out of all serogroups, just two are important, O1 and O139. The rest are not important organisms for medical microbiology. Of course, they can cause some disease, very mild diarrhea, for a couple of days and then it's gone. So nobody really cares about this. And they are called non-agglutinable Vibrio cholera organisms. Why non-agglutinable? Because O1 and O190 groups can be found by combining the culture of Vibrio cholera with a specific O1 or O139 serum. If it gives positive agglutination test, so we identify O1 or O139. So if it is negative, so the organism will be called non-agglutinable Vibrio because it cannot agglutinate with anti-O1 serum or anti-O139 serum. So nobody cares about it. So then, if we found O1 or Vibrio cholera with O1 somatic antigen from serogroup O1, then this serogroup organisms can be further subclassified according to the minor properties of cell wall into three serotypes: Ogawa, Inaba, and Hikojima. So, this is antigenic classification. But then apart from antigenic classification, based on some biological properties, Vibrio cholera from this O1 serogroup, you see here we all Vibrio cholera, and here we have Vibrio cholera from O1 serogroup. So it may be classified into two so-called biotypes, biological types. And they may be classified into classical biotype and Altor biotype. So, and the properties that allow this classification is susceptibility to specific bacteriophages, ability to produce hemolysis on blood agar, and some other biochemical tests. Disease. What is the pathogenesis of Vibrio cholera? It is transmitted through fecal-oral route, mainly water, and a bit rare food is the contaminated substance. The gastric acidity that occurs in stomach gives very good protection against disease. There's a very effective barrier. So the persons with achlorhydria or hypochlorhydria are very susceptible to the disease. Generally, the inoculum dose will be not less than 10 power 3. In the small intestinal tract, Vibrios cross the protective layer and reach epithelial cells within the help of high motility, then chemotactic adhesive pili, toxin coagulated pili, and mucinase that dissolve the mucin layer. So then, there they produce a cholera toxin. This is an exotoxin that is called choleragen. Why it is called choleragen? Because it generates cholera as a disease. All symptoms that we have mainly due to the activity of cholera toxin. And you can see here the cholera toxin. This is a Vibrio cholera that produces the toxin. Toxin binds with the special GM1 receptor by B subunits, and altogether we have 5 polymerized B subunits. And then you can see here the A subunit. And with the help of B subunits, it will be internalized by endocytosis into the cell. Here, the A subunit will be cleaved into 2 portions, and one portion will be very active. Then it will be released, the A portion will be released outside the endosome. And then it will interact with the G protein, and it targets adenylyl cyclase activated. It causes permanent accumulation of CMP, and as a result, hypersecretion of water electrolytes from the cell into the lumen of the gut. As a result, we have rice water stool, permanent diarrhea that cause hypovolemic shock. All symptoms that occur within the cholera as a disease is just because of the loss of fluid and electrolytes. So the major therapeutic strategy is to perform rehydration therapy. And it is even more important that just to give antibiotics. Of course, it is done, but rehydration is even more important than antibiotic treatment. So how do we perform the diagnosis? Traditionally, culture method is used. As soon as we get culture, we perform serological classification, biochemical test to identify the species, serological test to identify the sero group and serotype. Then we identify biotype, whether it is classical or Etor, and that will be the result of classification. But now quite often we may perform classification by polymerase, or we may perform diagnosis by polymerase chain reaction. And interestingly, the test kits are developed when you may identify several genes in the same set of polymerase chain reaction cycle in one tube. So usually we add primers for several genes. For the gene that code for O1 lipopolysaccharide antigen, this is the first pair of primers. Then second pair of primers is to identify toxin corregulated pillas. This is a very important virulence factor of Vibrio cholera. And the third pair of primers is added to identify the presence of cholera toxin. Then treatment is performed by rehydration therapy, and antibiotic treatment is also can be performed. So it may develop resistant to some antibiotics, but this is not very common for Vibrio cholera O1 serogroup. Several vaccines had been developed to perform specific prophylaxis or protection against this disease. So if you would like to travel to India, for example, of course this is not for Indian students. They had been there for a while, and there they get used to this. So, but the travelers supposed to be recommended to perform vaccination. And out of all vaccines, the one is considered more effective. This is so-called conjugative vaccine. So in this vaccine, the O1 lipopolysaccharide is conjugated with the cholera gene and a toxin or toxoid. Do you remember that this cholera toxin can be toxoided into anatoxin or toxoid? This is not toxic but immunogenically active substance, and if we use this conjugated vaccine, the antibodies will be developed in the vaccinated person against O1 somatic antigen first and against cholera toxin. So that is most protective. So protection cannot occur for very long while, but it may give several month antibodies. Okay, and the last disease we're supposed to discuss today, and I see that we won't have enough time to discuss it. This is Bacillus anthracis. It described not so bad in the lecture book, and you can see here the person who discovered it. And this is a photographer, Robert Koch, working in his lab. That is the one who discovered Vibrio cholera. Oh, sorry, Bacillus anthracis. Yeah, I'm confused already. Так. Out of all pathogenic bacilli, we are interested in Bacillus anthracis. This is principal antigen. And it had been isolated in culture by Robert Koch. And he showed that they have spores. They possess spores capable of producing spores. And Louis Pasteur produced the first attenuated vaccine done from bacteria in 1881. These two great men had some deal with the Bacillus anthracis. That is the picture of Bacillus anthracis organism. And that's how it appears in tissues. You can see that they are covered with a thin capsule, and they form so-called bamboo sticks because the organisms arranged in chains. So McFadden reaction may make the capsule more visible, and negative staining also can be used to visualize capsule, and the best way is to use direct immunofluorescent assay with the antibodies labeled with fits to demonstrate the capsule of Bacillus anthracis. And that's how it grows on the MPA, and you understand that this is not fastidious bacteria. It may grow on almost any medium. So capsule usually is not formed when we grow it on MPA. Capsule is formed in the human or animal body. So, but then if you want to demonstrate it in the laboratory, we have to add some serum, albumin or starch in a medium or bicarbonates. So, Bacillus anthracis produce spores. Usually they are central, ellipsoid. And if we perform gram staining, the spores are not stained. You're supposed to understand why. Because they are covered with so many layers, and it will be good if you know the names of these layers. So exosporium, spore coat, spore cortex or cortex, the remains of the cell wall of the core, then nucleoid and several ribosomes. That's what we have in spores. Almost no water, no metabolic activity, many layers cover it. So then, because of this, it is very resistant. Spores are very resistant to harsh conditions, and to get rid of spores, we can perform autoclaving. That is the best way to kill spores. It's not so difficult to grow Bacillus anthracis because it has very traditional cultural characteristics. But because they form the bamboo stick, so the edge of the colony will appear as medusa head appearance. So Bacillus anthracis cannot produce hemolysis on blood agar. Is non-hemolytic. And this is the way how we distinguish with some other bacilli, saprophytic bacilli. And it is biochemically active. It can ferment or decompose whole protein that we see in a column of gelatin. And strain of Pell reaction is used to distinguish Bacillus anthracis because it is sensitive to penicillin, and when we culture it on the medium with penicillin, the rods turn into cocci because the cell wall will be partially damaged, so and they will lose its osmotic rigidity. So if we culture it on penicillium medium for the longer time, so they will be ruptured. But if we subculture it on the medium without penicillin, they will revert to the original appearance of bamboo sticks. So this is example of the phenotypic variations of bacteria that depend on the environment. So, what is anthrax as a disease? Generally, it is a disease of herbivores, usually goat, sheep, cattle. Humans can be infected incidentally by contact with the dead animals or their products. So, but then Bacillus anthracis, because it is easy to culture it and it is very resistant due to the spores formation in the environment, is considered one of the most serious biological agents, potentially can be used as warfare agents. With this, I think we have to finish our lecture. So, and I hope you get an overview of some bacteria that can cause dangerous diseases. We will discuss some of them during the classes. And with this, I would like to wish you to have a very good weekend. And goodbye for now. So we are done with our lecture. Goodbye, my dears.